Hi all, welcome back to the DNA barcoding session. Welcome to the module 3 and the final session. So in the initial session, we have started with conventional taxonomy or traditional taxonomy and followed by the need of modern taxonomy. And module 3, we are going to discuss on the barcodes, markers used in barcodes, the repository of DNA barcodes, applications of DNA barcodes, and its scope and its limitation. And in module 2, the previous module, we have discussed about the definition of DNA barcoding, the father of DNA barcoding, and how DNA barcode came to existence, and what is the core need of DNA barcoding, followed by its methodology. One by one, step by step, we have seen. But here, I would like to give some inputs on the barcodes or the markers you use. So the last point, what we discussed in the previous module is the markers used in DNA barcode. So just to recap or to brush up, I repeat, for plants it was ribosco, for animals it is cytochrome C oxidase, and for bacteria or for prokaryotes it's rRNA, and for fungi it is ITS, that is the internal transcribed spaces. So here, the species identification and taxonomic assignment. So how we are going to exactly, you have isolated or you have pulled out the DNA, you have done some amplification followed by sequencing, and then you have given this to the software. So exactly what is happening. So if you have a sequence, now you have to compare or you have to do some reference with your library already which is existing. Then only you will be able to compare and you can tell, okay, this is the same one or this is a different one. So where these libraries are available. So what is the reference libraries or what is importance of reference libraries in DNA barcoding? In DNA barcoding, reference libraries are used for the taxonomical identification of sequences obtained from barcoding. So reference libraries is something we have to underline and we have to understand. There is a core area. The reference libraries are used for taxonomic identifications. Whatever sequencing you are bringing, that you have to compare with the reference libraries, and based on the comparison, after that comparison, you will be able to know whether it can be grouped in the same group or which belongs to a different group. Reference libraries contains databases of DNA barcodes assigned to previously identified taxa. So the previous data is well arranged in the reference libraries. Previously available, whatever database is available, that will be available in the reference library. Example, barcode of life data system, what we call it as BOLD. That is one reference library, and another one is UNITE, another reference library, and diet.barcode. And if you see a lot of molecular biology work in our country, in the reference articles or in the published article, we can see a lot of references from BOLD and diet.barcode. So example for the reference libraries are BOLD, UNITE, and diet.barcode. So here, after comparison, the comparison of the data which you have given and the comparison between the data you have given and the data which is already available in the tool. What's next? The fourth step is analysis by bioinformatics tool. As I told that for the computational biological well-established tools are available to obtain well-structured, clean, and interpretable data from the raw sequenced data. So here, when I'm talking about the output of the data, that means my claim is that the bioinformatics tool will give you a structured, clean, and interpretable data. So that is my wish, or everybody's wish who is going to do this DNA barcoding. But when you tell a clean data, and here it is mentioned as that raw sequence data you can give, but that doesn't mean that you have to give an unclean data. Whatever data you are giving, that should be also a clean pretty much. Then only the output data will be clean. So whatever outliers you are identifying, or if it seems to be a bit ambiguous, then again you have to repeat the process, because that will affect your output data. The output data will not be clean. So you should develop that expertise to avoid such outliers when you come with your sequenced data. So hope once you do this process, obviously available self-driven tutorials are available. So very easily you will be able to catch up the sequencing process, how it can be done with the help of a software. So now most of the software tools are versatile and highly user friendly. If you know how to use a Android phone in a better fashion, that skill will be more than enough to handle software which deals with sequence analysis and bioinformatics. Now, what I'm trying to tell means things are getting simpler. And here I would like to introduce some international bodies like International Barcode of Life. I think so. This was initially started. It is the largest biodiversity repository, or it is the to construct DNA barcode reference library. So still, if my knowledge is correct, and maybe it was one of the largest biodiversity genomics initiative to construct DNA barcode reference library. So please make a note of this body, IBOL, International Barcode of Life. Because as committed in module 2, I told that I will introduce some internationally renowned repositories to you. Canadian Center for DNA Barcoding, CCDB. Another barcoding repository, and it is claimed as the largest barcode factory. And what is the why I have included this slide? Apart from this faculty development or refresher interest, teachers who are planning to do their postdoc, or when you apply for sabbatical leave and planning for a postdoc research somewhere abroad, please approach such institutions in Canada, because as you know, unlike countries like US or UK, Canadian migration is something which is happening, and every day in our institution itself, we are signing at least 2 to 3 transcript for the students as well, the research scholars to apply in Canada, and they are getting visas in a pretty much easier fashion. So basically for botany teachers, because they usually train teachers and researchers in barcoding, unlike other repositories, Canadian Center for DNA Barcoding is a highly dedicated institutions for training, and there are a lot of opportunities are available, especially for the barcode researchers and for teachers. So please make a note of CCDB. It is again an out of interest. I have included this slide. So now again back to our presentation. What are the major applications of this DNA barcoding? Okay, so obviously we know that okay, the basic the major role is to identification of species. Okay, but still, if you delve into the details, okay, there are n number of opportunities and applications, interrelated applications, which are which is unseen by us. Okay, because we are only seeing the tip of the iceberg, only 20%, 80%. If you explore, lot and lots of applications are there. The first thing is that, as we mentioned, the primary duty is to identification of new species. Okay, because in morphological identification, again, you can't come to a conclusion just because of a color variation in the leaf, just because of the hairy growth on the stem, and just because of the color variation in the flower, because we don't know that such variations are whether stable or such variations are epigenetic. We don't know whether it is going to transfer to the next clonal descendants. We don't know. But such molecular level identification authentically based on the evolutionary relationship, based on the gene bank or based on their genome, you can conclude or you can tell whether it is a new species or not very easily. So that is the primary application of DNA barcoding. Safety assessment of food. So that we have not even thought about that concept, whether the food safety can be done here. So that can be done by barcode. Then detection of alien species. Okay, detection of alien species in the sense I'm not talking something extraterrestrial. If you such alien species or wide range of species can be identified here with DNA barcoding. So identification of endangered and threatened species. endangered species. Then the question comes on how you can identify endangered species, because they are not available in the earth, and how it is possible. Yes, from the fossil it is possible, from the fossil DNA it is possible. And if I'm going to some digging sites, some ancient civilization digging sites, if you are getting any DNA, or if you are getting any part of the hair, then we will be able to do a DNA barcoding. You will be able to pull out the DNA, amplify the DNA, anything can be possible if you have, if you give a picogram of DNA. I'm not even asking for a nanogram of DNA. If a picogram 10 raised to minus 12 picogram of DNA is available, then you can very well do a DNA barcoding. So endangered species it is possible. Even in threatened species it is possible. And even threatened species, again morphological identification is possible. But for quick identification, once it is endangered, everything is possible with the DNA barcoding, linking egg and larval stages to adult species. So that again in modern taxonomy, the scope is there, because in the module 2 we discussed about cladogram. This is this possibility is there, but still the fetal stages, the larval stages, maybe in multicellular stages, maybe in the stage of morula or whatever, maybe the linking the egg and larval stages, it is another revolution. It's another revolution where it is only possible with DNA barcoding, because as I mentioned, it is a non-invasive method. If it is an invasive method, then you will have to take the egg and you have to sacrifice, you have to take the larva, you have to sacrifice. So here it is no animal ethical issues, because you are not sacrificing or you are not killing anything, or you are not breaking a twig, you are not hurting a plant. So this is, or you are not sacrificing anything. So this is something which is the best part of DNA barcoding, which I want to reinforce to the teachers. So framing global management plans for conservation strategies, that looks to be a big generic term. Diet analysis studies, barcoding for food safety, in agriculture, in ecology, definitely in ecology, in environment studies, especially in evolutionary studies, in phylogenetic studies, this barcoding is going to revolutionize the entire system. So now potentials and shortfalls, because as I committed, the potential is time and cost, very quick, hundreds, thousands of sample can be done within days. Taxonomic resolution. Taxonomic resolution means you will be able to identify not only the species, even in strain level you will be going, individual organism level you will be going, minute variation you will be checking, strain level checking you will be able to done, especially in the case of prokaryotes, species identification. And again, high expertise is not required for this non-specialist. So usually we call a word called street smart. Okay, so street smart is something, for example, a person who is performing in a street, a person who is selling something in a street, he doesn't have any sole knowledge about the entire product or the entire process. The only thing is that he has to manage the show with the existing utilities or whatever it is available on that particular area. So you have to be only street smart, because you should know a bit of DNA identification, isolation. You should know how to amplify, and you should know how to sequence. Obviously, sequencing, a lot of bodies are there to give you the sequence. Only thing, you have to feed your data into the software, and analysis will be done by the system. Even using the artificial intelligence, the interpretation can be done. The only thing, based on your knowledge, you have to assess that whether our data is clean or not. And shortfalls are the physical parameters, definitely the collection of sample, and especially when you talk about endangered and all, some physical parameters are the shortfalls. Technological bias will be there, or technological limitations will be there, because the acceptance, how far you will be trusting the existing reference libraries. Though you call it as universality, in some part of the world it will be accepted, and another part of the world they may have a different school of thought in a particular species. So lack of standardization, because there is even universal barcodes are there, universal barcode markers are there. There is no universal repository for DNA barcoding. So that standardization will not be there. Each and every country or each and every region, they have set their own standards, but there is no uniform standardization for that. That may be a shortfall. Mismatch between conventional and barcode based identification, because I can't claim that somebody who is well expert in taxonomy for past 40 years and they are coming with a valid reason for based on the morphological thing, and you are telling that this is not based on the match, based on the genotype or its thing. So I don't think it is required that we should go for a debate on this score. We are not here for a open debate whether it is conventional or barcode or whatever it may be. And estimates of richness and diversity, and how the diversity and the richness of this scheme can be estimated, that all has to be debated and discussed. So that's it. So due to some technical reasons, I am just winding up this session, because in 3 modules or in 1 hour or 1 hour 20 minutes, we will not be able to completely talk about the entire DNA barcoding. But because friends, we live in a VUCA world, okay, or a VUCA DNA barcoding world. When I'm telling VUCA, we use CA because especially in when we talk about the shortfall, and V for volatility, and U for uncertainty, and C for complexity, and A for ambiguity. So always, whatever repository or DNA studies we will have, definitely we have this VUCA effect everywhere, VUCA effect everywhere. But overcoming all these shortfalls, we will concentrate on potentials, because this DNA barcoding is widely accepted, because due to its specificity, due to its distinctiveness, due to its high resolution, high taxonomical resolution between the 2 species, or even between the 2 strains, as I mentioned in some organisms, especially in the case of prokaryotes or bacteria or whatever it may be. Fine. So apart from this, I can share the freewares, softwares which are available. So I'm available in LinkedIn and Facebook, or I can share my email ID also. So I'll be more happy, because it is now a mono session. It's like a radio or a TV, because I'm just speaking. It will be broadcasted. So I would like to have this in an interactive way. So please raise your questions, and please feel free to contact, because always I'll be interested to network with people, especially with my colleagues and in the different part of the country. Okay, so that's it. So thank you Ramanujam College and Miranda House once again. It was a wonderful opportunity to share whatever small knowledge what we have to share with the teachers. So thank you all. So we will connect leisurely, and thanks for the opportunity. So it's me, Ayyappa Das, signing off. Thank you.